Imagine a plant that has been alive since before many world religions even existed,
Right.
surviving in one of the harshest environments on Earth, using absolutely nothing but the exact same two leaves it has possessed since the day it was born.
It's wild. I mean, it completely forces a total reevaluation of what biological senescence actually means, you know?
It really
We're
does.
basically looking at an organism that effectively treats its vegetative tissue as a permanent geological feature rather than like a disposable solar panel.
Yeah,
Mhm.
exactly. So we are metaphorically standing in the middle of the hyper-arid Namib Desert today, surrounded by just endless stretches of baking gravel and rolling dunes.
Sounds lovely.
Oh, it's a landscape that actively punishes complex life, yet it is home to one of the most astonishing biological anomalies on the planet, which is, of course, Welwitschia mirabilis.
The one and only.
Right, and for this deep dive, we've pulled together an incredible stack of sources.
We really have. We're integrating historical botanical records,
Yeah.
academic papers on desert ecology.
We've got the groundbreaking chromosome-level genome sequence that was published in 2021, right?
Yeah, that one is huge. And also rigorous recent radiocarbon dating studies.
Synthesizing all that data paints a picture of a species operating on a completely distinct evolutionary timeline.
Basically, yeah, it's a living fossil that just bypasses the standard rules of determinate growth and resource allocation.
So our mission today is to unpack the mechanics behind this anomaly. We're going to examine how Welwitschia rewrites the rules of biological aging, morphological constraints, and extreme survival.
Right, because to you, the learner listening right now, whether you are, analyzing complex systems in a corporate office or studying biology in a university lab,
The tactical trade-offs this plant makes to endure, well, they challenge our fundamental assumptions about resilience, resourcefulness, and like the actual cost of immortality.
I love that, the cost of immortality. To understand how Welwitschia sustains continuous cellular division for thousands of years, we have to start with its wildly chaotic, entirely anomalous physical architecture.
So weird looking.
It really is. It operates under a morphological paradigm that just abandons the conventional plant blueprint entirely.
Let's look at the historical context of its taxonomy first, because the sheer structural weirdness of it just stunned early biologists.
Oh, for sure. When the Austrian botanist Friedrich Welwitsch documented it in 1859.
Right, during an expedition in the deserts of southern Angola.
Yeah, the historical accounts actually suggest he fell to his knees in the desert sand.
Like he couldn't believe his eyes.
Exactly. He was convinced he was looking at a mirage. I mean, it looks like a pile of trash from a distance.
It really does. But I find the local nomenclature far more descriptive than the Latin name they gave it.
Oh, definitely.
In Afrikaans, it's Tweeblarkinidud, which translates to two leaves that cannot die.
Which is incredibly accurate.
And the Hero people call it Onyanga, which means the onion of the desert.
I love that one. Both names capture its ecological isolation and just bizarre morphology perfectly.
Taxonomically, its phylogenetic isolation is staggering, right?
Oh, yeah. As a gymnosperm, it sits within its own monotypic order, family, and genus.
It's all alone on the family tree.
Completely alone. Fossil records and molecular clock analyses place its divergence from its sister genus, which is Netum, at approximately 123.5 million years ago.
Wow, 123.5 million years. So it effectively locked in its evolutionary strategy during the early Cretaceous.
Right in the age of the dinosaurs.
It was already a distinct lineage when the continents were still, like, reconfiguring from the breakup of Gondwana.
Yeah, but the structural paradox is what really requires unpacking here, because the fundamental law of leaf development across almost all vascular plants dictates determinate growth.
Meaning they stop growing at some point.
Exactly. A primordium initiates, the leaf expands to a genetically predetermined size, and eventually, hormonal signals trigger abscission.
The leaf drops, like in autumn.
Right, the leaf drops. Welwitschia circumvents determinate growth through a radical ontogenetic shift early in its development.
Wait, what does that look like when it first sprouts?
So upon germination, the seedling produces two photosynthetic cotyledons.
Okay, the standard little seed leaves.
Yeah, and those persist for about 18 months. Concurrently, two true leaves emerge at right angles to those cotyledons from a specialized groove in the codex.
The codex being that woody base part.
Right, it's an inverted, woody, upconical stem structure. And at this critical juncture, the apical meristem, the central organizing tissue that dictates upward vertical growth in most plants, it just completely aborts.
It effectively decapitates itself.
Basically, yeah. By terminating its apical dominance, it prevents the formation of a trunk or a complex branching canopy.
So there are no branches, no twigs, no trunk reaching for the sky.
None of that. And the meristematic activity shifts entirely to the periphery of that woody crown.
Oh, okay, I see.
From that point forward, those two true leaves grow continuously from a basal meristem for the entire lifespan of the organism.
That's insane.
The actively dividing cells are localized at the base, pushing the older tissue outward horizontally across the desert floor.
Functionally, I mean, that is closer to the biological mechanics of a continuous extrusion process than traditional plant growth.
Oh, that's a great way to think about it.
It's like a highly sophisticated 3D printer that never pauses, just constantly extruding living, photosynthesizing biomass onto baking hot gravel.
Yeah, 3D printing leaves for a millennia.
But the visual reality in the field completely contradicts the two-leaf description. I mean, if you look at field photographs, they show giant, tangled heaps of green and brown ribbons.
It looks like a giant pile of green noodle.
Exactly. It looks like hundreds of separate leaves sprawling out from a central crater. So if it only has two leaves, why does it look like that?
Right, so that visual fragmentation is a direct result of environmental stress on the extruded tissue.
Ah.
I have this massive, broad, undivided leaf being forced continuously over abrasive desert pavement.
Over centuries, right?
Yeah, so over centuries, this sheer mechanical stress of aeolian sandblasting, whipping desert winds, and physical trampling by endemic megafauna.
Like rhinos and antelopes and stuff.
Exactly. All of that causes the tissue to split longitudinally along its parallel veins.
Wow. So the plant maintains vascular integrity along those individual strips, even as the broader leaf tears apart into a massive skirt of frayed ribbons.
Yeah, the plumbing still works, even though the leaf is shredded, and the scale of this extrusion is immense.
How big do they get?
We are looking at leaves that can stretch 4 to 6 meters outward, with the total circumference of a mature plant's canopy exceeding 8 meters.
That is massive for a desert plant. Driving a massive, continuous basal meristem engine to sustain meters of living tissue naturally demands immense metabolic energy.
Oh, for sure. But more critically, it demands continuous hydration.
Which introduces the primary hydrological paradox of this whole thing.
Right.
A continuous extrusion of broad, succulent leaves in the Namib Desert, a hyper-arid environment that averages less than 100 millimeters of rainfall annually.
Sometimes literally zero rainfall.
Yeah, it frequently goes years without a single drop. That violates basic principles of transpiration. How does it maintain turgor pressure without just depleting its internal water reserves?
Well, the answer lies in a highly specialized synthesis of atmospheric physics and photosynthetic modifications.
Okay, break that down for us.
So the Namib Desert's proximity to the Atlantic Ocean is the defining ecological variable here.
Yay, the ocean is right there.
The cold, northward-flowing Benguela current meets the intense thermal radiation of the desert landmass.
Hot air, cold water.
Exactly. This sharp temperature gradient generates a dense, persistent coastal fog that rolls inland, blanketing the desert for roughly 300 days a year.
Accessing atmospheric moisture is one thing, but retaining it under high thermal stress is another.
That's the real trick.
Right, because to run a standard C3 photosynthetic pathway, a plant must open its stomata during the day to fix atmospheric carbon dioxide.
Yeah, and in the Namib, the vapor pressure deficit during daylight hours is so extreme that a standard C3 plant would lose 97% of its water to transpiration almost immediately.
It would just instantly dry out.
It would be a crisp in an hour. So Welwitschia circumvents this through crassylation acid metabolism, or CAM photosynthesis.
CAM.
What sets it apart evolutionarily is that it is the only known gymnosperm to deploy CAM.
Really? The only one?
The only one we know of.
Yeah.
So during the peak thermal load of the day, Welwitschia keeps its stomatal pores completely sealed.
It locks the doors.
Exactly. It locks down its internal hydrology, entirely severing atmospheric gas exchange to prevent evaporative water loss.
Then it leverages the nocturnal temperature drop and the arrival of the coastal fog.
Got it. It opens its stomata at night to assimilate massive volumes of carbon dioxide.
And since the light-dependent reactions of photosynthesis cannot occur in the dark, it utilizes PPP carboxylase to temporarily fix that carbon into 4 carbon organic acids.
Primarily malic and isocitric acids.
Right. And those organic acids are sequestered within the large vacuoles of the mesophyll cells.
Yeah, it basically stores the carbon overnight, and as dawn breaks and the desert heats up, the stomata seal tightly shut once again.
Okay.
The plant then decarboxylates those stored organic acids, releasing the CO2 internally.
It saturates the Rubisco enzyme with this internal CO2, running the Calvin cycle, using the abundant solar radiation while remaining hermetically sealed against the arid external atmosphere.
It is a phenomenal closed-loop carbon partitioning system.
It really is. But the mechanics of how it acquires the water to maintain this system have been the subject of a massive paradigm shift in botany.
Oh, this part is fascinating.
Let's examine the stomatal density. The leaves are episomatic, meaning stomata are present on both the adaxial and abaxial surfaces, the top and bottom,
Right.
and they boast an incredibly high density of roughly 22,200 stomata per square centimeter.
Which is a huge number.
Right.
And for decades, that immense density led early botanists to formulate a very specific and, seemingly logical hypothesis.
They assumed the plant just drank the fog directly.
Exactly. They observed the plant bathed in heavy fog with millions of open pores on its upper surface and concluded that Welwitschia directly absorbed the suspended fog droplets through the stomata.
Functioning like a macroscopic sponge.
Yeah, that was the going theory for a long time.
But the fluid dynamics of that hypothesis always seemed problematic. I mean, pushing liquid water against the vapor pressure gradient through a microscopic pore designed for gas exchange, that is mechanically inefficient.
Yeah, the physics just didn't quite add up. And modern isotope tracing and hydrological studies have dismantled that early theory entirely.
The stomata are not acting as liquid intake valves.
No, they do not absorb liquid water directly. Instead, their primary function in this context is extreme hygroscopic sensitivity.
Hygroscopic meaning they react to moisture in the air.
Exactly. The stomata respond precisely to atmospheric humidity thresholds, opening in perfect synchrony with the arrival of the fog.
Primarily to optimize CO2 uptake when the transpirational risk is at its absolute minimum.
Exactly. The actual hydration mechanism relies on the macrostructure of the leaf acting as a passive condenser.
So the physical leaves themselves function as massive, waxy funnels.
Right.
The dense coastal fog condenses directly onto the vast, cool surface area of the leaves. As the microdroplets coalesce and gain critical mass, they follow the morphological slope of the leaf,
They roll backward toward the central caudex and drip directly into the soil.
creating a tight 1.5-meter radius around the stem.
It creates a localized, self-irrigating micro-catchment system. It's brilliant.
And below the surface, the root architecture is perfectly adapted to exploit this.
Yeah, well, Welwitschia does develop a deep taproot to access paleowater reserves. Its primary adaptation for fog harvesting is an incredibly dense, highly specialized lattice of upward-growing feeder roots.
Upward growing.
Yeah, negative gravitropic. These fine, spongy roots occupy the uppermost 30 centimeters of the soil profile.
Wow. They intercept and absorb that concentrated fog runoff immediately before the morning sun can evaporate it from the surface gravel.
It is an impeccably tuned system. Passive atmospheric condensation, coupled with active CAM carbon fixation and localized root interception.
But, engineering a system that works efficiently on a daily basis is fundamentally different from sustaining that exact same continuous cellular division for centuries.
Oh,
Biological tissues inevitably degrade, telomeres shorten, DNA accumulates replication errors.
Right, which forces us to confront the chronology problem.
Establishing the definitive age of Welwitschia individuals has historically been an analytical nightmare.
A total nightmare for botanists. You can't rely on standard dendrochronology because the basal meristem does not produce the distinct annual growth rings found in the cambium of temperate trees.
Right, the growth is continuous but agonizingly slow and lacks the seasonal cessation that creates a readable ring.
Yeah, without dendrochronology, early attempts relied heavily on mathematical extrapolation.
Like Hare's attempt in 1960.
Yes. Hare attempted to quantify their age by measuring the slow lateral expansion of the leaves and extrapolating that rate across the largest individuals on the Husab Plains.
And what did he find?
Well, he hypothesized that the massive specimens could exceed 2,000 years in age.
2,000 years.
Yeah, but, extrapolation over millennia is inherently fraught with variables.
We need empirical isotopic data, which brings us to the recent systematic radiocarbon dating study conducted on a dead, naturally sectioned specimen in the Welwitschia wash.
Near the Gobabeb Namib Research Institute.
The methodology here was rigorous. They didn't just extract a single core, they systematically mapped the isotopic age of the structural tissue across the entire transverse section of the caudex.
And the precision of the carbon-14 data they extracted is illuminating.
It really is.
They analyzed a relatively modest specimen, a dwarf tree with a meristematic circumference of only 226 centimeters.
So far smaller than the 8-meter giant.
Much smaller.
The C14 analysis, calibrated for the southern hemisphere atmospheric curve, dated the oldest intact tissue on that dwarf specimen to 531 plus or minus 20 radiocarbon years.
Which is incredible. That places the germination of this relatively small plant squarely between 1420 and 1440 AD.
To put that into a biological perspective, that equates to a vertical tissue accumulation rate of an astonishingly slow 0.47 to 0.67 millimeters per year.
Half a millimeter of vertical growth annually. Let that sink in.
It's glacial, but the isotopic mapping revealed a massive structural paradox regarding cambial growth.
Yeah, this is where it gets really weird.
Right. If you are analyzing a standard dicotyledonous tree, the oldest primary xylem is located deep within the central pith, while the most recent growth is pushed outward to the vascular cambium just beneath the bark.
Right, standard tree rings. But Welwitschia completely inverts that standard topological model.
How so?
Because of the anomalous basal meristem growth transferring to the periphery of the caudex early in its ontogeny, the oldest tissue is not protected deep inside the core.
It's not in the middle.
No. The isotopic mapping proved that the oldest carbon signature is located on the outer margins of the caudex tissue, right at the soil surface level.
And conversely, the internal cortex is significantly younger.
Exactly.
Structurally, that creates a massive vulnerability. I mean, if your primary cambial growth is inverted and your oldest structural tissue is permanently exposed on the exterior surface of the caudex, you are subjecting your chronological record directly to extreme aeolian erosion.
Sandblasting, basically. The implications for field researchers are profound.
I can imagine.
Technically, to sample the oldest tissue of a living Welwitschia, a botanist doesn't need to drive an increment borer deep into the core.
Which is good because that risks introducing lethal pathogens into an ancient organism.
Exactly. The oldest material is sitting right on the surface. However, the tragic limitation is that after centuries of severe sandblasting, extreme thermal cycling, and physical abrasion from desert fauna,
the absolute oldest exterior tissue on those 2,000-year-old giants has almost certainly been pulverized into dust.
Exactly. Yeah. The desert has essentially erased the physical evidence of their birth.
We might never isolate the true maximum age of the species because the material simply no longer exists.
It's gone, blown away in the wind.
But the fact that the genetic programming can sustain active, dividing, error-free meristematic tissue since the 1400s, or potentially the time of the Roman Empire for the larger specimens, without succumbing to rampant mutagenesis or cellular senescence, is staggering.
It's a biological miracle. And deciphering that genetic blueprint remained impossible until July 2021.
Right, when a massive genomic sequencing effort finally published a high-quality, chromosome-level genome assembly of Welwitschia mirabilis.
Unlocking that genome revealed an epigenetic vault optimized for extreme, prolonged survival.
Let's delve into the sequencing data. The genome is massive.
Huge. 6.8 gigabases, assembled across 21 chromosomes.
And hidden within that 6.8 gigabase sequence is a map of its deep evolutionary history and its response to paleoclimatic trauma.
Oh, absolutely. The genomic architecture clearly displays evidence of a whole genome duplication event that occurred approximately 86 million years ago.
Occurring squarely in the late Cretaceous.
Right. A whole genome duplication provides a species with a vast reservoir of redundant genetic material.
Because one copy of a gene can maintain essential biological functions, while the duplicate copy is free to mutate and develop novel functions.
Exactly. It drives rapid evolutionary adaptation. But the genomic trauma didn't end in the Cretaceous.
The subsequent paleoclimatic shifts drove the genome to expand further.
Roughly 1 to 2 million years ago, during the Pleistocene, the regional climate underwent severe, intense aridification.
The Namib was transforming into the hyper-arid system we see today.
Yeah, and in plants, extreme environmental stress, specifically intense drought and thermal shock, triggers massive bursts of retrotransposon activity.
Retrotransposons are mobile genetic elements. They utilize a copy-and-paste mechanism to duplicate themselves and insert those copies randomly throughout the genome.
Jumping genes.
Jumping genes. So this intense Pleistocene aridification triggered an explosion of these elements, heavily expanding the overall size of the Welwitschia genome by filling it with highly repetitive DNA sequences.
And the immediate biological question this raises is one of genomic stability.
Because if a genome undergoes a massive expansion of mobile, self-replicating elements, the statistical probability of those elements inserting themselves into essential coding regions and disrupting vital metabolic pathways is extraordinarily high.
Exactly. Over a 2,000-year lifespan, an unrestrained proliferation of jumping genes should theoretically result in catastrophic genomic instability.
It should trigger rampant insertional mutagenesis, effectively causing the plant equivalent of cancer.
Right.
How does Welwitschia maintain cellular integrity while harboring such a massive payload of retrotransposon?
Well, the 2021 sequencing project isolated the mechanism. It's an incredibly aggressive and highly specialized epigenetic restriction system.
Okay.
Welwitschia maintains extreme levels of cytosine methylation across its genome, specifically targeted at CHH motifs.
To conceptualize this for you listening, think of methylation as a biological read-only restriction placed on a software file.
Oh, that's a good analogy.
Right. The retrotransposon code is still physically present on the genomic hard drive, but the plant's epigenetic operating system has permanently revoked its execution permissions.
It locked it down. By heavily methylating the cytosine residues around these mobile elements, the plant structurally alters the chromatin, silencing the jumping genes and locking them securely in place,
It neutralized the internal genetic threat.
stabilizing its expanded genome to allow for multimillennial lifespans without mutational breakdown.
And furthermore, the genomic architecture is deeply optimized for the nutrient scarcity of the Namib.
Oh, this part is brilliant.
The DNA sequencing revealed that the Welwitschia genome is exceptionally GC poor.
Right. It has a significantly lower ratio of guanine and cytosine base pairs compared to adenine and thymine.
The metabolic economics behind this are fascinating.
Yeah.
Guanine and cytosine are nitrogen-heavy molecules. Synthesizing them requires a massive biological investment of nitrogen, which is a critically limiting macronutrient in the leached, sterile sands of the desert.
So by evolving a distinctly GZ-poor genome, Welwitschia fundamentally reduced the amount of nitrogen and metabolic energy required simply to replicate and maintain its own DNA.
It engineered a metabolically cheaper genetic code, perfectly suited for a nutrient-starved environment.
That is biological efficiency operating at the very foundation of life.
It's incredible. But, stabilizing the DNA and conserving nitrogen doesn't fully explain the physical persistence of the leaves.
Right, there's more to it.
Even with a stable genome, why doesn't the macroscopic tissue undergo programmed cell death? I mean, in virtually every other plant, sustained drought or extreme thermal stress triggers an unavoidable cascade of senescence.
The leaves degrade their chlorophyll, mobilize their nutrients back to the stem, and abscisse. They drop.
But not here.
No. The genomic study pinpointed the precise transcriptional module responsible for overriding that default biological programming.
And they designated it as the WMA1BF1-WMAYB111 module. Let's just call it the MYB111 module for short.
Good call. So the MYB111 transcriptional module functions as the ultimate longevity switch.
How does it work?
Under standard physiological models, when a plant detects severe stress, it synthesizes abscisic acid, or ABA.
This hormone acts as the primary signaling molecule to initiate senescence.
Exactly. But in Welwitschia, the WMMYB111 gene operates as a highly specialized inducible abscisic acid receptor.
So it intercepts the signal.
Yes. It intercepts and modulates the entire downstream signaling cascade. Instead of allowing the ADA to trigger the breakdown of chlorophyll and the cessation of cellular division, the MYB111 module fine-tunes the transcriptomic response.
It effectively silences the death signal.
It really does.
By strictly regulating the downstream genes involved in chlorophyll degradation and nitrogen remobilization, the MYB111 module artificially freezes the aging process of the young meristematic tissue.
It mandates that the basal meristem continues dividing and the leaf tissue continues expanding, utterly ignoring the environmental stress triggers that would force any other species into senescence and death.
It has constructed an impenetrable epigenetic and transcriptomic fortress against both internal mutational decay and external environmental trauma.
An absolute fortress.
But, as resilient as this isolated organism appears, ecological isolation is never absolute.
No plant is an island.
Right. Surviving for 2 millennia requires massive defensive adaptations, but propagating the species into the future requires deep, often vulnerable interactions with the surrounding ecosystem.
And this brings us to the profound ecological footprint of Welwitschia. Because it is one of the only massive, permanent shade-producing structures in the Namib, a single mature plant generates what ecologists classify as a fertile island effect.
A fertile island in a sea of gravel. It fundamentally alters the microthermodynamics of its immediate vicinity.
The sprawling, broad leaves create a dense canopy over the gravel. This intercepts solar radiation, dramatically lowering the soil temperature directly beneath the plant.
While the fog runoff we discussed earlier elevates the localized soil moisture.
Yeah, so in a landscape defined by lethal thermal extremes, this shaded microhabitat becomes an absolute magnet for endemic biodiversity.
It becomes a sanctuary. Desert-adapted reptiles seek refuge from the midday sun beneath the leaves. Small mammals occasionally gnaw on the tough fibrous tissue to extract emergency moisture.
And a complex community of arthropods utilizes the plant as a permanent foundation for their life cycles.
The most specialized of these arthropod relationships is with the Welwitschia bug, Probergrothius angolensis.
Yeah, the Welwitschia bug. This true bug is almost entirely endemic to Welwitschia populations.
It completes its entire life cycle on the plant, utilizing its specialized piercing mouth parts to feed directly on the plant's phloem sap.
But the relationship extends far beyond simple parasitism.
It's much more complicated.
It is a deeply complex push-pull ecological dynamic that exposes a terrifying vulnerability in the plant's reproductive cycle.
Let's talk about reproduction because Welwitschia is dioecious, meaning the male and female reproductive structures, the strobili, are housed on entirely separate individual plants.
Right. Males on one plant, females on another. And the spatial distribution of these individuals across the desert pavement requires a highly efficient vector to transport pollen.
From the male salmon-colored cones to the larger, strikingly blue-green or red female cones.
Exactly. They cannot rely on wind pollination. They rely heavily on entomophily.
Insect pollination.
Yeah. A suite of desert wasps, bees, and flies are attracted to the nectar exudates of the cones and facilitate cross-pollination.
And the Welwitschia bug is constantly traversing these cones, too.
Right. But its presence introduces a devastating biological variable. While the bug moves between the male and female strobili, it acts as the primary mechanical vector for a highly aggressive fungal pathogen.
Aspergillus niger var. Phonesis.
That's the one. The mechanics of the infection are brutally efficient.
How does it spread?
Well, the bug inadvertently gathers the fungal spores on its exoskeleton and deposits them directly into the microenvironment of the developing female cone.
Oh, no.
Yeah, the fungus then infiltrates the cone, sending its hyphae aggressively into the developing seeds.
It metabolizes the endosperm, rotting the seeds from the inside out before they even reach maturity.
Which systematically plummets the overall viability of the plant's reproductive output.
So the very micro-ecosystem the plant sustains actively sabotages its reproductive future.
It's tragic, honestly.
And even if a seed manages to escape the fungal pathogen, the environmental threshold required for germination is astronomically high.
The odds are just so stacked against it.
The seeds are morphologically adapted for wind dispersal, equipped with broad, papery wings designed to carry them across the dunes.
Dispersion across the arid landscape is successful, but establishment is nearly impossible.
For a seed to break dormancy, germinate, and successfully drive a taproot deep enough to survive the impending dry season, it requires a sustained meteorological anomaly.
It requires several continuous days of heavy soaking rain.
A climatic event that is fundamentally antithetical to the hyper-arid nature of the Namib.
Yeah, a multi-day soaking rain might only occur once every several decades.
And the consequence of this extreme germination threshold is visible in the population demographics.
When ecologists survey wild Welwitschia populations, they do not find a continuous gradient of ages.
You don't see 1-year-olds, 10-year-olds, and 50-year-olds all mixed together.
No. Instead, they observe highly distinct episodic recruitment events. They find wildly dispersed, even-aged cohorts.
You'll analyze one geographic cluster and find the entire population is exactly 400 years old, while another cluster 50 miles away might be uniformly 900 years old.
Because each cohort represents a single miraculous climatic window.
A year when the rains fell, the fungus was suppressed, and the seedlings managed to establish their root systems before the lethal heat returned.
It forces a critical analysis of their evolutionary trajectory, honestly. Welwitschia has achieved an unparalleled mastery of endurance.
Yeah, its morphological, hydrological, and genomic systems are flawlessly optimized to outlast extreme prolonged drought.
But its episodic recruitment rate, reproducing successfully, perhaps once in a century, creates a profound evolutionary lag.
It is the ultimate evolutionary tortoise. But that deep longevity masks a fragile inability to adapt to rapid environmental shifts.
If the ecological parameters change faster than its generational turnover, its localized extinction is highly probable.
As we synthesize the data from the morphology, the CAM photosynthesis, the genomic architecture, and the population ecology, a clear conclusion emerges.
Welwitschia mirabilis demonstrates that surviving the harshest extremes on Earth does not necessarily require metabolic dormancy, retreating underground, or shedding biomass like a deciduous tree in winter.
Right. Extreme survival can be achieved through a highly complex dynamic coordination of continuous, agonizingly slow growth.
It confronts the lethal thermal stress and nutrient starvation of the Namib Desert, not by retreating into dormancy, but by stubbornly, continuously advancing cell by cell.
Relying on a genetically locked down meristem and a perfect architectural utilization of coastal fog.
But as we project into the future, this living fossil presents a deeply complex ecological dilemma without a tidy resolution.
It really does. I mean, it survived the asteroid impact at the KPG boundary.
It survived the intense climatic fluctuations of the Pleistocene ice ages.
It survived the violent tectonic splitting of the Gondwanan supercontinent.
And it accomplished all of this by executing a singular, highly specialized strategy, growing the exact same two leaves for thousands of years.
But today, as anthropogenic climate change rapidly alters the precise oceanic temperature gradients that generate the life-sustaining fogs of the Benguela Current,
And as shifting thermodynamics accelerate the spread of the Aspergillus fungal pathogen,
that evolutionary strategy is being tested against an unprecedented rate of change. Its greatest strength, its stubborn, unyielding inability to change its specialized physiology, might ultimately become its fatal flaw.
it challenges our fundamental definitions of biological success. Does the attainment of ultimate millennial longevity inevitably demand a fatal sacrifice of evolutionary adaptability?
To the learner listening right now, thank you for joining us on this deep dive. The next time you observe a common houseplant rapidly dropping leaves or shifting its growth toward the light, consider the agonizing multimillennial endurance of the Welwitschia. Consider the staggering complexity required to simply stand still in a world that never stops changing.